Good afternoon, students. How are you all? So let us continue with the last discussion where we had discussed the structure of the two heme proteins, myoglobin and hemoglobin, starting with the heme group, right? The two were named as heme proteins because in both the cases the basic unit was consisting of heme group joined to a long polypeptide chain or the globin part. That's why both of these are placed under the category of heme proteins, right? Up to the last class we had taken a view of the structure of the two proteins. Today we have to check about the function of these proteins in our body, for which we will be requiring to discuss the topic dioxygen binding, transport and utilization, placed under another topic oxygen binding curve for the two proteins myoglobin and hemoglobin, or at the same time can be named as oxygen releasing or oxygen dissociation curve, or collectively can also be named as physiology of myoglobin or hemoglobin, right? So for that purpose we have to study this diagram, this graph in between percentage saturation of the two proteins myoglobin and hemoglobin, and on the x-axis taken is the concentration of oxygen in terms of partial pressure of oxygen. Why? Because oxygen is a gas, right? So let us start with this discussion. So to start with, as you know that in our body, this we have already explained, हमने पहले से explain किया हुआ है that in our body the heme proteins myoglobin and hemoglobin carry out the function of bringing oxygen to cells of the body, where oxygen is used in cellular respiration or various metabolic processes to produce energy rich ATP molecules. The collective function of the two proteins is to bring oxygen from lungs to the tissues to perform various metabolic activities for production ultimately of the energy rich ATP molecules. Now what is the structural difference? To recall again that in hemoglobin and myoglobin, heme groups are present which are responsible for binding of oxygen, where what is the difference between the two proteins? In myoglobin we are having the presence of only single heme group, therefore it can bind one oxygen, whereas at the same time hemoglobin can bind 4 times more oxygen because of the presence of 4 heme groups in its structure, right? myoglobin में एक हीम प्रेजेंट है तो एक ऑक्सीजन को बाइंड कर सकता है, हीमोग्लोबिन में 4 हीम ग्रुप्स प्रेजेंट हैं तो वो 4 गुना ज़्यादा ऑक्सीजन को बाइंड कर सकते हैं, right? Next, now as we have discussed that the function of both the proteins is to carry oxygen from lungs to tissues or muscles, right? Then the question arises, if the function of both the proteins is same, why we require the two different proteins to bring about the same function of carrying oxygen, right? Or you can say that why our body is needing two different proteins for the same purpose. This is because these two proteins differ slightly in their structures. The answer to this question lies in the fact that the two proteins, these are slightly differing in their structures, which is producing a slight difference in the properties of these two proteins, and hence a slight difference in the function of the two proteins, right? So because of this small difference in the structure of the two proteins, the myoglobin, this is actually used as a storage house for oxygen, whereas hemoglobin is the one which brings oxygen continuously from lungs to tissues, right? So the two proteins, although performing the same function but with a slight difference, that myoglobin used actually as a storage house of oxygen releases oxygen only when required, whereas hemoglobin brings oxygen continuously from lungs to tissues, releases the carried oxygen to tissues continuously, right? Now to describe these facts we need to explain the oxygen binding or oxygen dissociation curve, right? Or you can say to describe these properties or functions of the two proteins, the functions that we have discussed already, that to describe these properties or functions of the two proteins we require oxygen binding or oxygen dissociation curve to be discussed, right? So if now you have to pass through the curve first. The curve, as again I had told you, that this is in between percentage saturation of the proteins, myoglobin or hemoglobin, and partial pressure of oxygen. This is actually concentration of oxygen, as oxygen is a gas, therefore represented as partial pressure of oxygen, and is given in the unit torr, where 1 torr, this is equivalent to 1 millimeter of mercury, right? Here we have drawn the two curves, where you can see from the graph the different nature of the two curves for myoglobin and for hemoglobin. Where the curve for myoglobin is simple hyperbolic in nature, the curve for hemoglobin, this is sigmoidal or S-shaped, right? So what the difference in these curves is indicating, let us see it for myoglobin first. That for myoglobin we have observed a simple curve showing that as the concentration of oxygen increases, the curve it shows a sharp and quick rise, right? Means as the concentration of oxygen increases, the curve rises sharply and quickly. For example, let us take a point on the x-axis which is standing for concentration of oxygen. Let us take 2 millimeter of mercury. At this point, approximately this point, this is standing for 20 millimeter. This point will be approximately 2 millimeter of mercury. You can see the binding tendency of myoglobin towards oxygen. Means at 2 millimeter of mercury, myoglobin, this will be 50% saturated, or 50% of the myoglobin, this will be saturated with oxygen, right? Means at a very small concentration of oxygen, myoglobin immediately accepts oxygen and thus shows a strong affinity for oxygen. Means I have shown here the point that 50% of the myoglobin, it gets saturated with oxygen only at the very small level, a very small concentration of 2 millimeter of mercury, right? So what this indicates, it indicates 3 points basically. First, myoglobin binds oxygen very quickly. Second, myoglobin has a strong affinity for oxygen. Third, myoglobin will not be releasing oxygen until partial pressure drops to a low quantity, right? So I have given you 3 points. The first 2 points, these are standing for acceptance or binding of oxygen, whereas the third point, this is standing for the release or dissociation of oxygen, right? So remember, the same curve, it can be used both for binding and dissociation. If we move on the right side of this curve, this will be showing the binding tendency of the two proteins towards oxygen, and if we move on the opposite side, on the left, this will be showing the releasing or dissociation tendency of the two proteins for oxygen, right? So both properties, that is binding nature or dissociation nature of oxygen for the two proteins, can be discussed by the same graph, right? So next, now I have given you 3 points. The first, oxygen is binding quickly, strong affinity for oxygen by myoglobin, and third, myoglobin will not be releasing oxygen until partial pressure drops to a very low value, right? So all this happens, all the 3 points, these will be possible because of the structure of myoglobin, which is consisting of a single heme group and single polypeptide subunit, right? And because of this, not showing the cooperativity effect, right? Remember that cooperativity effect we had discussed for hemoglobin, where in the structure of hemoglobin we were having 4 heme groups present, interacting with each other, making space for the next oxygen molecule to bind with the units, right? Means cooperativity effect, which is shown by hemoglobin because of the presence of 4 heme groups, is not shown by myoglobin because of having a single heme unit in the structure, right? Thus the myoglobin, this will be unloading oxygen when partial pressure of oxygen drops to a very low value, right? Remember that by the same curve, we are not only discussing oxygen binding tendency, but also oxygen releasing or dissociation tendency of the two proteins, right? Next, now, so what is the physiology for myoglobin by the curves? What we obtain from the curves, what is the physiology? for myoglobin that our body can use myoglobin for oxygen storage until it really needs it. राइट जब तक ऑक्सीजन की ज़रूरत एमरजेंसी में नहीं पड़ती, बहुत ज़्यादा ज़रूरत नहीं पड़ती, मायोग्लोबिन का क्या रोल होगा उसको अपने पास सेव करने का. राइट सो when as I told you that our body can use myoglobin for oxygen storage until it really needs it. Right, when the cells have very low oxygen, or this will be possible in those cases when the cells these will be having very low oxygen, or when the lungs these are not able to release oxygen to tissues effectively. Right, so when myoglobin will be playing its parts, this will be done in the case when the cells these are having a very low oxygen concentration, or the lungs these cannot release oxygen effectively to tissues. Right, next, now this is the basic nature of the myoglobin hyperbolic curve, the simple curve, and the function actually of myoglobin to act as the storage house for oxygen. Right, next let us discuss the sigmoidal or S-shaped curve for hemoglobin, or oxygen binding or oxygen dissociation curve of hemoglobin. Right, so now we are moving towards the curve for hemoglobin. Unlike myoglobin which is not sharp, and is sigmoidal or S-shaped in nature. Right, so this sigmoidal or S-shaped curve for hemoglobin, this will be indicating that the binding affinity is much small in hemoglobin than in myoglobin. Right, you can see the nature that binding, where the binding tendency of myoglobin for oxygen was very high, the binding tendency for oxygen by hemoglobin this will be very small. Right, so this is the first difference in between the two proteins by their curves, which is explained by the nature of their curves. Right, next, now for example, as I have told you that myoglobin this will be having very strong affinity for oxygen, hemoglobin at the same time will not be having such a large affinity for oxygen. Right, this can be explained by taking the same 2 millimeter mercury concentration. Right, so we are using the same example that we had used for myoglobin. For example, a concentration of 2 millimeter of mercury. Right, remember, a concentration of 2 millimeter of mercury, it was enough to bind 50% of myoglobin to oxygen. Right, so again I am saying a concentration of only 2 millimeter of mercury is needed to bind 50% of myoglobin to oxygen, and comparative to this, if you move by the same point onto the hemoglobin curve, comparative to this, a concentration of approximately 26 millimeter of mercury, right, a concentration of 26 millimeter of mercury will be needed for hemoglobin molecule to bind oxygen. Right, where only 2 millimeter was required to saturate 50% of myoglobin, 26 millimeter will be required to saturate 50% of the hemoglobin. Right, hemoglobin. Right, so this is showing that hemoglobin it has a very low affinity for oxygen, and also it releases oxygen molecule readily as compared to myoglobin. ठीक है, remember that myoglobin it was having quick tendency or strong affinity to accept oxygen, strong affinity for oxygen, and was hard to release it. Right, whereas for hemoglobin it has less affinity for oxygen and can release it easily as compared to that released by myoglobin. Right, both of these effects are related oppositely to each other, more binding, less releasing. Right, so this is the difference in between the two proteins. Right, so next, now to recall again, hemoglobin carries oxygen from lungs to tissues or muscles or cells of our body, where myoglobin is used to store oxygen in the muscle cells. whereas myoglobin is used to store oxygen in the muscle cells, right? So this is the same function that I have repeated, that hemoglobin is used to carry oxygen from lungs to tissues, whereas myoglobin is used to store oxygen in the muscle cells, right? Next, now why we have obtained the S shape or sigmoidal curve in case of hemoglobin? This is because of the nature of the structure of hemoglobin, that this is having 4 heme groups in its structure, right? Sigmoidal curve for hemoglobin is obtained because of its structure, where in the structure it is having 4 heme groups present, right, which show the cooperativity effect, right? So, or you can say the S-shaped curve for hemoglobin is observed as a result of binding of oxygen to hemoglobin in a cooperative manner, right, where the different heme groups interact with each other towards binding of oxygen, which means that binding of oxygen at one site of hemoglobin makes the other unoccupied site more likely to bind oxygen, right? Binding at one site of hemoglobin makes the other unoccupied site more effective, more likely to bind oxygen. Or oppositely, you can say conversely, the release of an oxygen from one site on hemoglobin makes the other occupied site more likely to unload oxygen, to release oxygen, right? So this is about the basic nature of the two curves and about their functions. Myoglobin not releasing oxygen quickly, therefore is used as the storage house of oxygen. Releasing only when required, means in case of emergency, when the partial pressure of oxygen is very small, whereas hemoglobin, this will be quickly releasing oxygen, or the function actually of hemoglobin is to carry oxygen from lungs to tissues or muscles, right? So next, now we have to move towards the opposite side of this curve, as we have to check for the transportation of oxygen in from lungs to tissues to perform various activities. Means we will be requiring the oxygen dissociation or oxygen releasing curve to be explained under this phenomena, right? So for that purpose, I am moving towards your notes that I had supplied to you, that I had given to you to read about, right? So in this case, as per the oxygen releasing or oxygen dissociation curve, we have to start with the partial pressure of oxygen in lungs, right? We have to start from the upper hand side of this curve, which is showing the partial pressure of about 100 millimeter of mercury, and this is prevailing in lungs, right? So in lungs, as you can imagine, that when oxygen this is entering into our body, this is through the respiratory tract. ऑक्सीजन हमारे नाक से या मुंह से अंदर जाती है, रेस्पिरेटरी ट्रैक से वो सीधा लंग्स में पहले पहुंचती है, राइट? सो व्हाट विल यू बी सेइंग अबाउट द पार्शियल प्रेशर ऑफ ऑक्सीजन इन लंग्स, दैट दिस विल बी वेरी हाई, राइट? सो सेम इज शोन in this case, that in lungs the partial pressure of oxygen, this will be around 100 millimeter of mercury, and you can see the nature of the two curves, the upper myoglobin, lower hemoglobin, that both the curves, these are having almost same very high affinity for oxygen, right? Means at this point when oxygen is entering into lungs, both the proteins myoglobin and hemoglobin, these will be having a very high affinity for oxygen and will be binding to oxygen effectively to produce MbO2 or HbO2 and 4, right? Means dioxygen complex of the two proteins will be formed immediately in the lungs, right? Next, now from the lungs through blood, this oxygen will be carried into the tissues or muscles to carry about to perform different body functions, right? So when we move along the curve on the opposite side, remember that when we are moving on the opposite side, we are discussing the dissociative nature or releasing nature of oxygen, right? So when we move from lungs towards the muscles, I have taken this region in this curve, which is in between 20 to 40 millimeters of mercury, which is the prevailing pressure of oxygen in muscles, right? So as compared to lungs, this partial pressure of oxygen in muscles, this is very small, and you can see the difference, the differing nature of the two proteins as far as their curves are concerned, right? So here in this case, from the curves you can get an idea that where hemoglobin, this is having a very low affinity for oxygen, myoglobin still is having a large affinity for oxygen, right? So what this is indicating, that at a very low partial pressure of oxygen prevailing in muscles, hemoglobin, this will be having less affinity for oxygen, but myoglobin still be having more affinity for oxygen. So what this will be indicating, that hemoglobin having low affinity for oxygen will tend to release oxygen when comes into muscles or tissues, right? And this oxygen released, this will be accepted by myoglobin having more affinity for oxygen at the same partial pressure, right? That we had already discussed, that even at a low partial pressure of oxygen, myoglobin is having a strong affinity for it, right? So myoglobin in muscles, this will be immediately accepting oxygen to produce MbO2 and will be releasing oxygen only in the case of emergency, means when partial pressure of oxygen supplied by the lungs is very small, right? So this is what we have written in those cases. Apart from oxygen release from hemoglobin into the muscles or tissues, the other important function which is carried out in the tissues, that tissues, these are needing oxygen in great amounts, right? Means need for oxygen is greater in tissues which have already consumed oxygen and have produced carbon dioxide. You all know about the function of oxygen, that this is used for metabolic processes to produce carbon dioxide at the same time, which through the same cycle will be exhaled from our body, right? So oxygen not only is released by hemoglobin and absorbed by myoglobin, but at the same time oxygen is used for various metabolic functions to produce carbon dioxide in our body, right? Now what is the role of this carbon dioxide produced? Carbon dioxide produced, this will be reacting with water present in our body to produce carbonic acid, which immediately will be dissociating into H ions and bicarbonate ions, right? So what you can make a relation, which relation you can make, that more is the carbon dioxide produced, more will be the H ion concentration in our body. What this increased H ion concentration will be bringing about, that as the H ion concentration increases, it will be decreasing pH at the same time, right? So where this will be relating to that H ion increases acidity, thus decreasing pH, and this increase in acidity will again be favoring release of oxygen from HbO2 to myoglobin, right? Means two important factors have to be noted: low affinity for oxygen by hemoglobin in muscles at a low partial pressures, low affinity for oxygen by hemoglobin at a low partial pressure, and large production of carbon dioxide and hence H ions and hence decreasing pH. Both of these factors, the low affinity for oxygen at low partial pressure and high production of carbon dioxide and H ion concentration, both factors, these are affecting release of oxygen from hemoglobin to be absorbed by myoglobin, to be reacted by myoglobin, right? So this is all that we have to discuss up to this point. The second factor which is also playing its part, that is the production of carbon dioxide and hence H ions, increasing the release of oxygen from hemoglobin towards myoglobin. This is named under Bohr's effect. This will be discussed under Bohr's effect in the next topic, but this is about the oxygen dissociation curve that we have explained. And next, moving to the another flow chart which was given into your notes, let us come across this flow chart which is given into your notes. We have divided this flow chart into three regions: the lungs, blood and muscles. In the lungs, oxygen will be inhaled at a very high partial pressure of oxygen, that is 100 torr or 100 millimeter of mercury. This will be reacting with hemoglobin. Hemoglobin in lungs at high partial pressure, strong affinity for oxygen, so will be accepting oxygen very quickly, and this HbO2, the dioxygen complex of hemoglobin, means this will be carrying oxygen through blood and will be moving towards the muscles, right? So oxygen inhaled combined with hemoglobin into the lungs, carried through blood into the muscles or tissues where very low partial pressure of oxygen is there, which is showing low affinity for oxygen of hemoglobin and high affinity for oxygen of myoglobin, right? So in muscles, the partial pressure of oxygen is small, hemoglobin affinity for oxygen will be small, but myoglobin affinity for oxygen will be large, right? So at this low partial pressure prevailing in muscles, hemoglobin, which will be having low affinity for oxygen, will immediately be releasing oxygen that will be accepted by myoglobin having strong affinity for it, right? And this myoglobin turning to the dioxygen complex of myoglobin will be acting as the storage house and will be releasing oxygen only for the metabolic processes when required, right? Now this oxygen, by after passing through the metabolic processes through cellular respiration, will be producing carbon dioxide, which again passing through the blood will be exhaled from the lungs, right? So this is all about oxygen binding curve that we have explained today. This is very important. And next, what is your exercise for today? That you have to go through this topic, oxidation and oxygenation of hemoglobin and myoglobin. From this topic you will be coming out to a conclusion that you have to study as an exercise for today. You have to study about the role of globin part in hemoglobin or myoglobin, right? So this is all for today. If you get any problem, you can contact me anytime anytime you want, right? So this is all for today. Good luck. Go through the topics and rest. Bohr's effect we will be discussing in the next lecture. Thank you.